Very good morning, friends. Today we are recording this video for the student of TY, and we are, as usual, studying the genetics. As we know, the Mendel, figure George Mendel is the father of genetics, and we have gone through his life history as well as the laws which was postulated by him, that is a law of dominant, law of independent assortment, and the law of segregation. Now we move forward to study a monohybrid ratio. So what is the monohybrid ratio? What is hybrid? Hybrid is the outcome of one pure and a different allele. Now, in this hybridization, we study monohybrid as well as dihybrid crosses. Now, how do we get this monohybrid crosses? Monohybrid is in this cross, the result we find is that three of are the pure and one is the hybrid in phenotype or in the genotype. So what is the relationship between the genes and the characteristics? Genes are the genotype, and the character which we observe are the phenotype. Phenotype is equal to genotype plus environment. Well, genetically identical, growing in soils of different acidity, you can look out the flowers, the bushy flower which are along the wall. Phenotype is equal to genotype plus environment. Principle applies equally to the human characters. What are the primer? Genes come in a pair that separate in the formation of gametes. The members of the pair may be identical or non-identical. When the members of the pair are identical, then they are to be known as the homozygous, as well as when they are non-identical, then we may call them as a heterozygous. Each form of a particular gene is an allele. Well, only two alleles of a given gene are possible in an individual, although many alleles of a gene are possible within a population. One allele is dominant over another, or so Mendel believed. Genes, alleles, and chromosomes. Possible pairing of a homologous chromosomes are shown in these diagrams. Well, you see the three pairs of the chromosomes. Each pair consists of a maternal as well as the paternal chromosomes. Well, you are aware of the structure of the chromosomes, short arm and long arm. Both are connected with the centromere. Look out at the boxes which are colored. One is shown with the yellow one and the another one is shown with the green one. Whereas the yellow one represent the dominant alleles and the green one represent the recessive alleles. So friend, what we see in the homozygous dominant or in the homozygous chromosomes, we see the genes present at the same loci and are of the same color. Look out at this. So when you see here, see, this is the genes which are located on the yellow bar one. See this one, you can observe this. This is the homozygous dominant, whereas the reproduction, what we get here are the yellow seeds. Now, next to the heterozygous one, look out at the heterozygous one. Though the seed color is yellow, but the genes is the heterozygous one. This one is the green one and this one is the yellow one. Well, when we move towards the homozygous recessive, see, you find the homozygous in both the condition, is in dominant as well as in the recessive. What you see in the homozygous recessive, both the genes are located on the same position with the green color, and the product of these genes, or the product of the genes and the character that is represent, that one is the green seed. So these things, keep this thing in mind, and you can clear your doubt very clearly regarding the homozygous gene, regarding the heterozygous gene, regarding the homozygous dominant and homozygous recessive. So this is the location of gene for a seed color. See, the location of the gene is present on the chromosomes on the same loci, that are the homologous chromosomes. When the genes are present on the same loci of the chromosome, then they are termed as the homologous chromosomes. But what about the homozygous dominant? In homozygous dominant, you see the maternal and paternal both in the same color on the same loci, whereas on the heterozygous, one in the dominant representing yellow color and other which is representing the recessive one. Though the dominant one shows the phenotypically yellow color, but the recessive gene is present over there, but it is not expressed due to the expression of the dominant one. Whereas in the third pair, that is of the chromosomes, you see the both the genes present on the chromosome, that the genes are of recessive one, and that is why it is shown of the in the green color. Okay, you see the 3 seeds are shown here, 2 are of yellow seeds and 1 of the green seeds. Well, this is the flower of the pea, that is the Pisum sativum one. You look out here, this one is the flower, male anther, and you see the ovary over here, this is the stigma, female, and the seeds or the capsule you see in the yellow seeds are over there. Now after sowing the seeds in the soil, the germination takes place. Now we move towards the texture of the one we have seen regarding the color of the genes in the monohybrid one. Now we move for the texture of the seeds. See, look out these pods. This is the Pisum sativum pod, that is round and wrinkled, 2 alternative traits of the seed shapes character. See, when we see the same shape character on a individual plant, on a same plant, a pod of a individual plant shows some seeds that are wrinkled and shows some that are smooth in the texture. So two alternative traits of the seed shape characters are observed over here. Note that each of seed is a new individual of a different generation. Seeds are not the same generation as the plant that bears them. Now we go for the monohybrid cross. Look out at the monohybrid cross. For selection of the monohybrid cross, so in this diagram or in this slide, you can observe the monohybrid cross that is shown over here. See the F1 cross. In yellow seed, those are taken from the female one and the green one are from the male one. Well, as the abbreviation of the genes, that is genotypes, there are both capital Y1. And in case of the green one, green color, the genes that are of smaller y1. While putting or while crossing these two, what we get the possible outcomes in the fertilization. A Punnett square, something we will cover in a moment. See, this is the Punnett square, and female gametes and the male gametes are distributed in a reciprocal form. When the female gametes are put from the left hand side from this way, and the male gametes are put from the on the opposite sides. When capital Y and the smaller y are crossed among themselves, then the product is this one, that is the capital Y and the smaller y. So capital Y and a smaller y, when they are fertilized, the possible outcome comes that I am zooming out this so you may find this clear one. Look out at this capital Y is fertilized by the smaller y. What the genotype is shown in the yellow one, that is the capital Y and the smaller y. So you can see the F1 generation with these four seeds. Now this F1 generation is fertilized among themselves. They are allowed to grow in the plant. Okay, you can see the F1 generation plants are grown over there and they are self-fertilized. So staying the course, Mendel continued crosses to the F2. What was learned, the green trait was not lost or altered even though it disappeared in the F1. Though yellow was dominant, one trait is dominant to the other in its expression. So you can observe here in this slide, yellow one is the dominant in the while crossing over there. That means the female genotype is dominant over the male genotype. Well, while crossing self among themselves, what we see in the F2 generation, that is this. See, look out the genotype. When we got the dominant one, yellow one, we sowed them into the soil. The plant arises with the flower, and whatever the seeds we get from them are here put in the Punnett square. Well, the from the left hand side you can see the female gametes, which is shown over here. This is the female gametes and this one is the male gamete. When in the next generation, that is in the F1 generation, while dominant Y for the yellow color was crossed with the male Y, then we got both the genotype, that our genes are capital Y, but in case of the color they are shown as a yellow. Well, look out here, we see the 3 yellow and 1 the green, that is 3 is to 1. But what about the genotype? What we see in genotype? We see in genotype that one seed is shown with the capital Y, both the letters are capital Y, and 2 seeds are shown with the capital Y and a small y, but phenotypically they are yellow in color, and one, that is green one, pure one, is shown with the smaller y. That is, we see 1 is to 2 is to 1 genotype, and the phenotype what we see is to 3 is to 1. The reappearance of the recessive traits in is 1/4 of the F2 generation. That suggests genes come in pairs that separate in the formation of sex cells. What happens here in F1 generation, while we allow the pure parents to reproduce, then we get the 4 seeds with the pure yellow dominant one. That means what we see here that the female is dominant over male one and male is the recessive over female one in F1 generation, because no green color has appeared in this generation. What in the next, after fertilization, after self-fertilization of F1 generation, of first filial generation, what we get in F2 generation? We get in F2 generation that the pure characters, that is yellow one and green one, are expressed, but though dominant one is more one. The recessive allele which was suppressed in the F1 generation reappears in F2 generation, but though it reappears, it shows phenotypically, but the number of seeds is more of dominant one, that is 3:1 ratio. So this was what about the monohybrid cross and the principle of the segregation. Well, we'll look out in this diagram. So friends, today we have learned regarding, with the help of the experiments of the Mendel's and the Punnett square, how he segregated the genotypes and the phenotypically characters while crossing the pure parents and adopting the hybrid one and self-fertilizing this hybrid one, and how he got the ratio, that is the 3:1 in the phenotype and 1:2:1 in the genotype. Well, we stopped here, and rest of the part of our this monohybrid crosses will be going to study in our next lecture. Till then, thank you. Enjoy your day.